Good afternoon. The title of this presentation is Cortical EEG RIMS in Neurodegenerative Disorders. I have no conflict of interest here. Dementia reflects severe cognitive deficits and disabilities in the activities of daily living. This presentation focuses on neurodegenerative progressive disorders such as Alzheimer's disease and dementia with Lewy bodies. According to international guidelines, the diagnosis of Alzheimer's disease can be done with biomarkers, such as the measures of beta amyloid and tau in the brain. In contrast, the diagnosis of Lewy body diseases are based on clinical features together with some biomarkers. Our general scope is to introduce EEG biomarkers in the research framework of the National Institute of Aging and Alzheimer's Association. The guidelines by Jack and colleagues in 2018. You can see in the slide the amyloid, tau, and neurodegenerative biomarkers used in the research framework. And we are working to convince the people to introduce EEG biomarkers between the biomarkers of tauopathy and the biomarkers of neurodegeneration. Amyloid and tauopathy are based on biomarkers of cerebrospinal fluid and positron emission tomography. And the neurodegenerative biomarkers are based nowadays on anatomic magnetic resonance imaging and fluorodesoxyglucose positron emission tomography. Ideally, EEG biomarkers of brain functions could detect patients with Alzheimer's disease and Lewy body diseases from preclinical stages without cognitive symptoms or clinical manifestations to dementia. Among EEG biomarkers, we used the EEG activity related to the resting state condition of eyes closed and open. It's a very easy and a diffuse approach to study neurophysiological mechanism of vigilance. EEG alpha rims of quiet vigilance were introduced by Hans Berger in the beginning of the last century. You can see in the red framework the EEG activity recorded during eyes closed resting state condition. You can see the alpha rims, ample oscillations in the posterior areas at the frequency of about 10 Hz. During event-related activity, the alpha rims disappear and you can see the beta rims emerging in the EEG traces. EEG rims reflect the synchronization of cortical neural activity. You see that specifically the pyramidal neurons in the cerebral cortex have the spatial orientation of the main dendrites, allowing the summation of postsynaptic potentials. And this summation is able to reach the scalp electrodes and allows the recording of EEG. Synchronization means the similar timing of a neural activity in the cerebral cortex. And you see that when you have a similar timing among neurons, you can have a summation and the recording of waves in the EEG activity. In contrast, when the activity of neurons in the cerebral cortex are not synchronized, you have a flat EEG activity at the scalp level. The EG alpha rims in the resting state are prominent in the posterior scalp regions and the posterior cortical areas. They reflect cortical inhibition in relation to no visual stimulations or auditory stimulation and psychophysical relaxation. In the slide, you see the model of cerebral cortex and thalamus, which may generate EG activity and alpha rims. This model was validated in animal models with electrodes implanted in the cerebral cortex and in the thalamus. The EG alpha rims may be generated by reciprocal loops between thalamus and cerebral cortex, modulated by ascending activating system, particularly with using acetylcholine as a neurotransmitter. So alpha rims may reflect a traffic light function of this thalamocortical loop. EG activity is a complex signal that can be decomposed in elementary oscillations at different frequencies. And this allows to identify the individual alpha frequency peak, the specific frequency in the alpha range showing the maximum amplitude. And you can see in the slide how you can represent the amplitude of the power of single EG oscillations in the EG power spectrum. So what do resting state EEG rims tell us about Alzheimer's disease and Lewy body diseases? In our studies, we addressed the hypothesis that resting state EEG markers may reflect cortical neural synchronization in a different way in Alzheimer's disease and Lewy body diseases. And that the resting state EEG markers may be related to disease clinical manifestation of these diseases. Our general approach is based on the recording of a resting state, eyes closed and eyes open conditions with the 1020 electron montage system using 19 electrodes. So it's a low resolution for the EEG activity. And we used artifact-free EEG periods as an input for exact low-rate freeware, allowing the estimation of cortical sources of EEG rims. You can see that we use large regions of interest in relation to this low resolution of EEG recordings. This is the consortium Pharmacog, a European consortium investigating the biomarkers of Alzheimer's disease in patients and animal models. Some studies were performed within the framework of the Pharmacog project. The first study of the Pharmacog project I present today is related to the validation of a resting state eyes-closed EEG biomarkers in alpha and delta RINs in relation to Alzheimer's disease neuropathology as revealed by cerebrospinal fluid, A beta and phospho-tau biomarkers. We compare the EEG source activity in Alzheimer's disease patients with mild cognitive impairment as opposed to mild cognitive impairment patients not due to Alzheimer's disease. You see the sources and the activity of the sources for different frequency bands in the horizontal axis, the intensity of cortical activation in the sources in the vertical axis. In the red, you have the power spectra of source activities for Alzheimer's disease and mild cognitive impairment. And in blue, you have the same spectrum of cortical source activity for MCI people not due to Alzheimer's disease. You can see that even the two groups show the same cognitive impairment. You have a difference in the source activity, especially in the alpha RIN, in parietal, occipital, and temporal areas, with significant effects in the parietal and the occipital areas. Another effect can be seen in the delta source activity in the temporal areas. In general, you see that Alzheimer's disease patients with mild cognitive impairment show lower alpha source activity as opposed to MCI patients not due to Alzheimer's disease. So the general level of posterior cortical inhibition in the resting state eyes closed condition is different in these two groups of patients. Another validation of resting state EEG markers was performed by the correlation between global EEG source activity and brain hypometabolism biomarkers of positron emission tomography. The study was performed in Alzheimer's disease patients with dementia, and you see a significant correlation between global delta source activity and the brain hypometabolism. The higher the delta source activity in the cortical sources, and the higher the brain hypometabolism in the typical regions related to RD neurodegeneration. In the same line, we validated EEG biomarkers during the resting state condition and the cortical gray matter volume in MCI and dementia patients with Alzheimer's disease. You see in the graph that the higher the global delta source activity and the lower is the cortical gray matter volume, as revealed by structural magnetic resonance imaging. In contrast, the higher the alpha source activity in the cerebral cortex, and the higher is the cortical gray matter volume. So in both cases, we have a nice correlation, significant correlation between delta and alpha source activities and the cortical gray matter volume, as revealed by magnetic resonance imaging biomarkers. Of course, you can see variability at the individual level. Any circle here is a subject, but anyway, there is some statistical correlation between these variables. Then we studied the resting state, eyes closed EEG biomarkers in Lewy body diseases in the framework of PD Waves Consortium and European Lewy body disease consortium. You can see that several researchers contributed to these studies, and in red you see the researchers more active in the EEG module of scientific activities in these two consortia. In one of the studies, we compared the resting state EEG biomarkers in normal elderly subjects, in blue in the graph, Alzheimer's disease with dementia in red, Parkinson's disease with dementia in green, and Lewy body disease with dementia in violet. Again, you see that in the vertical axis you have the cortical activation in sources. In the horizontal axis, you have the frequency bands from delta to gamma and the cortical regions of interest in the source space. Again, the normal elderly subjects show the most prominent alpha source activity in the posterior cortical regions of interest, in the parietal and occipital areas. You can see in blue the maximum values of cortical source activities. And in the diagram, you can see that the Alzheimer's disease patients with dementia show the lower level of alpha source activity. A dramatic drop of alpha source activities in the parietal, occipital, temporal, and limbic regions of interest. The Parkinson's disease with dementia and the Lewy body disease with dementia in green and violet show a significant reduction of alpha source activities, but not so strong as Alzheimer's disease patients. So alpha source activity in the posterior areas was a nice biomarkers of neurophysiological differences in the regulation of vigilance in these patients. Furthermore, you can see that in the delta range, we have differences among the groups. You can see that normal elderly subjects show relatively low delta source activity in occipital areas, in the parietal areas, in the temporal areas, and that there was an increase of delta source activity in Alzheimer's disease in red, but the greater increase in delta source activities was observed in Parkinson's disease with dementia and Lewy body disease with dementia. So alpha source activities and delta source activities in the resting state as closed condition show differences in these groups. So we tested the hypothesis that this kind of differences in alpha and delta ranges could be observed also in the prodromal stage of dementia, the mild cognitive impairment condition. And so again, you see that we compared normal elderly subjects with Alzheimer's disease patients with mild cognitive impairment in red and Lewy body disease patients with mild cognitive impairment in green. Even in this study, you show that the prominent alpha source activities were observed in normal elderly subjects in parietal and occipital regions of interest. And that the lower alpha source activity was found in patients with Alzheimer's disease and the mild cognitive impairment. Furthermore, we observed that also Lewy body disease patients with mild cognitive impairment show a decrease of alpha source activity, not in the same measure of Alzheimer's disease patients with mild cognitive impairment. Finally, we could replicate that even in mild cognitive impairment patients, the increase of delta source activity in several regions of interest in patients with Lewy body disease and mild cognitive impairment. A significant increase of delta source activity was observed also in Alzheimer's disease patients with mild cognitive impairment. So even in the mild cognitive impairment stage, you can see the typical picture of EEG biomarkers observed in dementia stage. The same kind of comparison was performed between Alzheimer's disease patients with mild cognitive impairment and Parkinson's disease with mild cognitive impairment. And the results were very similar to the other study comparing EEG biomarkers in Alzheimer's disease, mild cognitive impairment, and Lewy body disease with mild cognitive impairment. Here, Alzheimer's disease patients are in red, Parkinson's disease patients are in green, and normal elderly subjects are in blue. And the picture is very similar to the previous study. Alzheimer's disease patients with mild cognitive impairment showed the lower levels of alpha source activities in different regions in the posterior areas, and we had the intermediate level in the Lewy body disease patients, and also some greater delta source activity in Lewy body in Parkinson's disease patients with mild cognitive impairment. So we can see here the spectrum of Alzheimer's disease and Parkinson's disease. In the left side, we have Alzheimer's disease with A-beta amyloid and tauopathy, and the typical Alzheimer's disease related genetic risk factors. In the right side, we have the alpha-synuclein pool in Parkinson's disease patients and the typical Parkinson's disease-related genetic risk factors. Halfway, we have a Lewy body disease with the genetic risk factors similar to Alzheimer's disease and Parkinson's disease, and also a beta amyloid profile in some cases, and of course, alpha-synuclein accumulation. You see that Alzheimer's disease pool is most strictly related to alpha source activity, a reduction of this variable, and Parkinson's disease, an increase in delta source activity. And Lewy body disease seems to have a mix of EEG features of Alzheimer's disease and Parkinson's disease. So we investigated the relation between EEG biomarkers and clinical manifestation in Lewy body disease. And in this study, we compared EEG biomarkers in the resting state, eyes closed, in Lewy body disease patients with greater cognitive deficits in red and Lewy body disease patients with lower cognitive deficits. And you see that patients with higher cognitive deficits show an increase of theta and alpha source activity in parietal and occipital areas. No differences in delta source activities. When compared to this circumscribed effect in the theta and alpha, what we observe comparing Parkinson's disease patients with higher cognitive deficits and the lower cognitive deficits, you see greater cognitive deficit in green and lower cognitive deficit in red. You see that there is a specific increase in alpha, but not theta and delta, and this increase was diffused in all regions of interest, so widespread when compared to the previous circumscribed effect in Lewy body disease patients. In Parkinson's, is widespread and limited to alpha. In this contrast, we compared Lewy body disease people with visual hallucinations in red and no visual hallucination in blue. And the effect was specific for delta range in the parietal region of interest, with greater delta source activity in Lewy body disease patients with visual hallucination. So very circumscribed and related to delta range. A very different picture was observed in Parkinson's disease with visual hallucinations compared with Parkinson's disease patients without visual hallucinations. Here the visual hallucinations were represented in green profiles of source activity. And you see that we have, as opposed to Lewy body disease, that Parkinson's disease with visual hallucination have a higher alpha source activity in parietal, occipital, and temporal, so in the posterior regions, quite widespread, and no effect in delta range. So we could see in the spectrum across Alzheimer's and Parkinson's disease that Lewy body disease and Parkinson's disease show a different profile of EEG biomarkers in the resting state, eyes closed, in relation to cognitive functions and visual hallucinations. An increase of delta, theta, and alpha source activity in Lewy body disease patients, and a more circumscribed and limited effect in alpha source activity in relation to cognition and visual hallucination. In conclusion, delta source activities are more abnormal in Parkinson's disease and Lewy body disease patients, whereas alpha source activities are more abnormal in Alzheimer's disease patients. And these effects were observed in both mild cognitive impairment and dementia stages. Concerning Lewy body disease patients, the cognitive functions were related to theta and alpha source activities in these patients. In contrast, In Parkinson's disease patients, the effects were observed in alpha source activities. In the same line, visual hallucinations were related to delta source activities in Lewy body disease patients and alpha source activities in Parkinson's disease patients. So we have nice evidence that EEG biomarkers show different neurophysiological mechanisms in Alzheimer's disease, Lewy body disease, and Parkinson's disease patients. So you see my co-workers in the Roman group, and these are the European funds supporting this research. Thanks a lot for your attention.